Howdy, my name is Jazlyn Gonzalez and I am part of group number 6 with Andres Garza. Today we will be going over Biochemical Test Lab 3. In this lab, we were able to identify and differentiate bacterial species based on their enzymatic activity and biochemical reactions using biochemical tests. The four major tests performed included the catalase test, oxidase test, nitrate reduction test, and antibiotic susceptibility test, each targeting specific characteristics that help distinguish between bacterial genera and species. The catalase test detected the presence of the enzyme catalase, which breaks down hydrogen peroxide into water and oxygen. The formation of bubbles indicated a positive test result, meaning that the organism can protect itself from the harmful effects of reactive oxygen species. The oxidase test identified bacteria that contained cytochrome C oxidase, an enzyme involved in the final step of the electron transport chain. A positive result, shown by a color change on the swab after adding the oxidase reagent, indicated the organism uses oxygen for energy production through aerobic respiration. The nitrate reduction test determined whether an organism can use nitrate as a final electron receptor during anaerobic respiration. After incubation, reagents are added to detect nitrite or nitrogen gas formation. A red color change would indicate a positive result, meaning nitrate was reduced to nitrite. If no color change developed, zinc was added to confirm the result, where if the broth turned red after the addition of zinc, the test was negative. The results of this test show whether the organism can reduce nitrate to nitrite, nitrogen gas, or further, demonstrating its respiratory versatility. Lastly, the antibiotic susceptibility test helps determine how effective specific antibiotics are against different bacterial species. Discs containing antibiotics such as bacitracin, novabiocin, and optochin are placed on inoculated agar plates. The presence or absence of a clear inhibition zone around each disc reveals whether the bacteria are resistant or sensitive to the antibiotic. Now for our materials. For media, we used 5-nitrate broth Durham tubes, 4 blood agar plates. For cultures, we used Alcaligens faecalis, Escherichia coli, Pseudomonas aeruginosa, Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus saprophyticus, Streptococcus pyogenes, Streptococcus agalactiae, and our unknowns number 136 and unknown number 276. For equipment, we used glass slides, H2O2, petri dish, cotton swabs, oxidase reagent, butane tank as our aseptic heat source, and an inoculating loop.
As for our procedure, we have three different tests for this lab experiment. Starting off with the catalase test, we have number one, a colony was transferred to a clean glass slide, followed by a few drops of 3% hydrogen peroxide that was added, and 3, the reaction was observed for bubble formation indicating oxygen release. As for the oxidase test, we're starting off with a sterile cotton swab that was used to collect a bacterial colony, and then 2 to 3 drops of oxidase reagent were added. 3, a dark purple color within 30 seconds indicated a positive result. Now for the nitrate test, each organism was inoculated into nitrate broth with Durham tube and incubated. After incubation, sulfonylic acid and alpha naphthalamine were added. And 3, if no color developed, zinc was added to detect unreduced nitrate. For 4, results were interpreted based on color change or gas production. Now for antibiotic susceptibility test. For 1, blood agar plates were inoculated with a uniform lawn of bacteria. For 2, bactitracin and optochin discs were placed on streptococcus plates. Novabiocin and staphylococcus plates. And 3, after incubation, zones of inhibition were measured to determine sensitivity or resistance. So now moving on to our results and observations for the catalase test. We're going to first start off with Staphylococcus aureus. So in this observation, we noticed that there was an immediate bubbling after adding hydrogen peroxide, which resulted in a positive test. And this then interpreted to the we could then interpret this into meaning that there is a presence of catalase enzyme, which basically breaks down hydrogen peroxide into water and oxygen, hence the reason why there's bubbling. Now for Streptococcus pyogenes, our observation was that no bubbles were seen, meaning that this was a negative result, and this was primarily due to a lack in catalase, which means that it was not able to decompose the hydrogen peroxide. Now for unknown 276, we noticed that there was vigorous bubbling upon hydrogen peroxide contact, meaning that it was a positive result, indicating that it was a catalase positive organism and possibly a Staphylococcus species. Now lastly, for unknown 136, we saw that there was no visible bubbles after hydrogen peroxide addition, meaning that it was a negative result, indicating that it was a catalase negative organism and possibly a Streptococcus species. So now for the results and observations with the oxidase test. We have 4 different types of tests that were conducted. However, I just want to mention that for E. coli, we did 2 different tests because we noticed that on the first test, there was an immediate appearance of purple. However, we retested it and there was no appearance of purple. So we believe that this is the correct one since E. coli is supposed to lack cytochromatin, C oxidase, meaning that it's a facultative anaerobe. This also results in a negative test, and it's also the reason to why no color change happened after the reagent. So initially, the first color change was probably just some kind of error. However, for P. aeruginosa, the swab immediately turned dark within about 20 seconds, indicating that it was a positive test result. And for the interpretations, we identified that this contains cytochrome C oxidase, meaning that it's an obligate anaerobe. Now, for unknown 136, we noticed that there was no color change that was detected, meaning that it was a negative result. And for the interpretation, this essentially meant that it does not produce cytochrome C oxidase. Now, for unknown 276, we did notice that there was a slight color change in purple. However, it was just very little, and this essentially means that it was a positive result, meaning that it is likely an anaerobic organism with oxidase enzyme.
Now for the results and observations of our nitrate test. From left to right, we have E. coli, which was deemed negative result as there was no color change after the reagents and the zinc were added. Next, we have P. aeruginosa. This test was also deemed negative as there was, again, no color change after adding the reagents and the zinc. Third, we have A. faecalis. This test was deemed positive as there was an immediate red color change after adding the reagents. There was no need to add zinc. Next, we have our unknown number 136. This test was deemed partial positive as it did turn red on the lower half of the tube after adding the reagents. Again, we did not have to add any zinc to this test. For our unknown number 276, this was deemed negative as there was no color change after adding the reagents and the zinc. Now for the results and observations of our antibiotic susceptibility test. Starting with S. saprophyticus, we saw a clear ring around the antibiotic novobiocin, which tells us that this bacteria is susceptible to this antibiotic as it was able to inhibit the growth of the bacteria in this region. The mean diameter of this ring was 1.78 millimeters. Next, for S. epidermidis, we saw a clear visible ring around the antibiotic novobiocin. This also told us that this bacteria was susceptible to this antibiotic as it was able to inhibit the growth of the bacteria in this region. The mean diameter of this ring was 2.23 millimeters. Next, for S pyogenes, we saw a clear ring around the antibiotic bacitracin, but not around the antibiotic opticin. This told us that the bacteria is susceptible to the antibiotic bacitracin as it was able to inhibit the growth of the bacteria in this region. The mean diameter of this ring was 1.27 millimeters. Lastly, for S. agalactiae, we saw no clear rings around the antibiotic bacitracin or opticin. This told us that this bacteria is not susceptible to these antibiotics and couldn't inhibit the growth of the bacteria in these regions.
So, in conclusion, this lab demonstrated how biochemical and antibiotic susceptibility tests can be used to identify bacteria based on enzymatic reactions and drug resistance. Each test provided diagnostic insights to catalyze and oxidize identified metabolic pathways, nitrate reduction, revealed respiration type, and susceptibility testing help distinguish similar species, crucial for both clinical and microbiological applications.
Below is a list of our references that we used for the procedure and for our introduction.
So this concludes our lab report video presentation, and thank you all for listening.